Experiment 23, the detection of fats, proteins, and carbohydrates in foods. The purpose of this experiment is to introduce you to some general tests which detects fats, proteins, and carbohydrates in foods. So Part A, test for detecting carbohydrates. So the first test we're going to do is the Molish test, and this is a general test for carbohydrates, and any monosaccharide will give a positive test. Since the test solution is acidic, disaccharides and polysaccharides will hydrolyze to give monosaccharides, so they too will give a positive response. So we're going to prepare and number 5 15-centimeter test tubes as directed below. 20 drops of DI water, 20 drops of 1% glucose solution, 20 drops of 1% sucrose solution, 20 drops of 1% lactose solution, and 20 drops of 1% starch solution. So to each of the 5 test tubes, we're going to add 3 drops of the Molish reagent and then seal each test tube with a cork or film and shake each tube to ensure mixing. Then with the test tube inclined at a 45-degree angle, slowly add 10 drops of concentrated sulfuric acid so that the 2 layers form. Use caution when handling concentrated sulfuric acid and keep the mouth of the test tube pointed away from yourself and others around you. So note the color produced where the 2 layers meet the interface and record your observations on the report sheet. A purple color is a positive test for monosaccharides, and you should see this with the glucose sample. So the next test is the iodine test for detecting carbohydrates. Polysaccharides can combine with iodine to form a blue, red, violet, or purple complex. So we're going to prepare 5 test tubes with fresh samples of the carbohydrate solutions used in the first test. And then we will add 1 drop of iodine solution to each and shake to mix. Observe the control sample, which is the DI water. Okay, so we added the 1 drop of iodine solution to each, and this one showed a positive test. But if you did not have any samples that had a different color than the control, then you can add 10 drops of pure water to each test tube and then 1 more drop of iodine solution. And you'll want to make sure to mix each solution well and then record your observations on the report sheet. And starch will give a positive test, which is what we got. And the next test is the Benedict test for detecting carbohydrates. So before proceeding, set up a boiling water bath in a beaker half filled with water, and then add boiling chips to ensure smooth boiling. Any carbohydrate that can form a free aldehyde group in a solution will react with the Benedict reagent to form a brick red, brown, or occasionally yellow precipitate. A precipitate of any of these colors is considered a positive test. However, a color change in the solution without the precipitate is a negative test. So we will prepare 5 clean test tubes with the same samples of the solutions used in the other 2 tests, and then we will add 5 ml of the Benedict reagent to each test tube and shake to mix. So remove the 5 test tubes after 5 minutes in the hot water bath and record your observations on the report sheet. And just remember that any carbohydrate that can form a free aldehyde group in a solution will react with the Benedict reagent to form a brick red, brown, or occasionally yellow precipitate. A precipitate of any of these colors is considered a positive test. However, a color change in the solution without the precipitate is a negative test. Part B, test for detecting protein. So the first test we're going to do is the xanthoprotic test. When treated with concentrated nitric acid, the aromatic rings on the side chains of proteins or free amino acids become substituted with nitro groups. The nitrated products are yellow, so the solution becomes yellow or a yellow precipitate forms. When the solution is made basic with sodium hydroxide, the color becomes more intense and ranges from yellow to orange to brown. Prepare and label 6 15-centimeter test tubes with the following solutions: 20 drops of DI water, which is the control, 20 drops of 1% biuret solution, 20 drops of 2% biuret solution, 20 drops of 2% tyrosine solution, 20 drops of 2% egg albumin solution, and 20 drops of 2% glycine solution. To each of the 6 solutions, add 5 drops of concentrated nitric acid and handle this with care. Then we will heat the solution in a boiling water bath for 3 minutes and record the colors produced on the report sheet. So we heated the solutions in a boiling water bath for 3 minutes. And now we will remove the tubes and allow them to cool. And then we will cautiously and slowly add 15 drops of 6 molar sodium hydroxide to each of the test tubes. And then you will want to stir each with a glass stirring rod and remove a droplet of each solution and touch it to a piece of pH paper. Add about 5 more drops of sodium hydroxide to any solution that does not test strongly basic, and recheck the pH and add more sodium hydroxide if necessary until each solution tests strongly basic with pH paper. Record any color changes on the report sheet. And tyrosine will give a positive test. So we were able to get some color changes in some of the test tubes, and now we will test it with the pH paper. And that one was able to show a basic solution. So the second test we'll perform is the biuret test for detecting proteins. So we obtain fresh samples of the solutions listed in the Xanthar product test in 6 15-centimeter test tubes. To each, we will add 5 drops of 6 molar sodium hydroxide and 3 drops of 2% copper sulfate solution. Seal each test tube and shake each mixture. And then after 3 minutes, we will note the colors produced.